name is Joerg Albert. I'm a reader in sensory neuroscience at the UCL Ear Institute. And I'm studying there the molecular basis of mechanosensation with a particular focus on hearing. We use model organisms because in my lab we carry out basic research which we'd like to try to translate in the end to human applications or clinical stages. I'd like to know how on the molecular level mechanical senses work and what the hell they are used for. We have lots of different mechanical senses. We know the sense of touch, we've got the sense of hearing, the sense of gravity. And all these, of course, are part of certain disorders and pathologies. So directly relating to that is the question, can we heal these disorders? Can we cure these pathologies? But much more my interest is on the basic level of research. I want to know how the molecules that bring about these sensations interact and what the molecules are in the first place. When reassessing our findings so far, I'm always surprised by how diverse the findings are in contrast to the original simplicity of the questions we asked. So what, for example, we found that I'm still, every time I'm thinking about it, I'm surprised about it. We know that every animal has a sense of time. And this sense of time is based on an endogenous autonomous oscillator which is called the circadian clocks. Circadian clocks operate like molecular feedback loops which maintain a 24-hour rhythm, but which have, so they can tell you the length of day and night, but they can't tell you when it's day and when it's night. In order to have that information, they need external sensory clues, and those are usually light-darkness clues. So light-darkness rhythms are required for all circadian clocks in unicellular organisms or in our own species in order to inform the circadian clock. We know from many other species that temperature is also required, or sufficient partly, in order to entrain, as you say, as to synchronize the clock. So in contrast to the sensory pathways for light, we had no idea at all what the sensory pathways or requirements for temperature are. So we found several years ago that a type of sensory organ called chorotonal organ, which are mechanosensory cells, are required for the fly's entrainment to temperature cycles. They can mean two things. Either the previously called mechanosensory organs are actually also temperature sensors, or temperature-dependent entrainment is not a temperature sensing process, but it's a mechanical somehow sensing process, which could be explained, for example, by assuming that a temperature leads you to be active. The activity is measured by mechanosensors. So essentially it's a mechanosensory phenomenon. Then we tried to see, can you replace the temperature entrainment by mechanical entrainment? So vibrate the flies. And surprisingly we found that vibration is enough to entrain the clock. This, however, directly relates to mammalian species. Mammalian species such as ourselves have been reported to be influenced in their timing, in their rhythms, in all disorders with regard to their circadian behaviors by activity. So if you are active or not active, it's relevant for clock and clock-related diseases. This, for example, they try to exploit this for activity-based therapies in clock-related diseases such as depression. But without knowledge about the sensory pathways and the right timing of these therapeutical approaches, these will never work out. So this led from fruit fly-based research to potential implications in human and mammalian species. In this research, again, we found that clock genes, so the genes that form the core molecular components of the biological, the circadian clock, are expressed also in ears, but they don't serve any clock function there. What we have seen so far is that eliminating them leads to a hyperamplification of the ear, something that in humans may be called tinnitus. So perhaps clock genes contribute to regulating our auditory sensitivity. Again, the fruit fly research has to be validated in mammalian species, but in the fruit fly so far, over the past 10 years, we have established lots of different strategies how to analyze mechanosensory ion channels. And this is something that in fruit flies is possible, but very difficult to establish in more complex mouse-based, for example, model organisms and systems. Like to many things, to answer why my research particularly or the research we are conducting in the lab is so important to society has at least two different answers. The one is a very concrete, specific one, and the other is a general one. Generally, I would say the research that we are doing is basic discovery-driven research, which accounts for the fact that our knowledge of the world stands in stark contrast to our perceived knowledge of the world. We know so much less about the fundamental processes than we think we know, that basic research's prime task is to do away with this overconfidence. We do not know enough to answer most of the questions. Even when we are writing the papers, we answer very specific ones, very specific questions, never the big ones. The big ones are the umbrellas. So, as a prime example of basic research, I think from an educational and cultural point of view, the research we are doing, especially at a faculty of neuroscience at UCL that is so world-renowned and that is offering facilities and techniques which are unavailable in many other parts of the world. We play a cultural and educational role in informing the public of essentially how little we know. And the second, more specific question, of course, is we are looking into basic sensory mechanisms, how the sense of hearing comes about, how the sense of gravity comes about, how the sense of touch comes about. And this molecular inspired knowledge could inform new pathways of treatments for lots of diseases, ranging from depression, if we look at the clock genes. We also have research in circadian clocks. It can be research into the treatments of tinnitus, so this phantom sensation that leads to ringing of the ears in the absence of any sound, which can be quite devastating for those suffering from it. And of course, treating or preventing hearing loss or deafness is one of the major tasks of this institute, and we feel committed to this translational approaches. But again, we are more committed to finding the questions and translating the questions to other model organisms than already finding the treatments or the therapies. But first comes the question, then comes the answer.